Good day. In this lecture, we will be discussing about Introduction to the Bioinformatics, Accessing Sequence Data, and the Literature. So this is the first lecture in our course, Bioinformatics 1. So just a brief reference and copyright notice. So the reference used in this presentation are from Bioinformatics and Functional Genomics by Jonathan Pevsner. So some of the images are gotten from this book. Okay, so a brief outline of this lecture. So it will be a series of lectures or lecture videos with this key outlines. So we will discuss about introduction, a brief introduction to bioinformatics, and some key bioinformatic websites, and then what are the biological databases that we can use, and in particular, the one that is most used is NCBI. So what is NCBI, or National Center for Biotechnology Information? So, first, let's discuss bioinformatics inherently is a tool. It's a tool to study biology, to explain biological phenomena. So, let's say, for example, we have a question. We want to know or we want to analyze a protein. So, using bioinformatics, how do we analyze the proteins? So, for example, we can have hemoglobin. So, we are interested in learning about the beta hemoglobin, or we call it HBB, hemoglobin B, beta. So which is a subunit of hemoglobin. So when we say a subunit, it's part of the protein. So we have the hemoglobin, which is a large protein. So it has 4 subunits, 2 beta and 2 alpha. So in particular, we're looking at the beta subunit. So we want to know about that. So we can use the NCBI, the usual go-to for most people. It offers information on the levels of the DNA, so we can know about some variations in the gene. So what can these variations mean? So it can sometimes point to diseases and some variants of diseases. So we can have RNA data. So RNA, as you know, is an expression. RNA is the molecule that is synthesized when the gene is being expressed. So we have expression data. So if we want to know about the expression of certain genes, we look at the RNA, not the DNA. We look at the DNA if you want to we know about the sequence of the gene, if there are any mutations that can cause diseases. So that's where we will use the DNA. If you want to know the expression, we go to the RNA. And then what about the protein? So we have proteins, protein analysis, so we can have the sequence of the protein, or we can know about the 3D structures of the protein. So we have protein databases, and a more meta version of that is looking at the pathways where our protein is connected. So these are for NCBI. Alternatively, we also have other databases. We can look at EBI. So we have comparable information actually in another, it's somewhat like NCBI, but this one is the European version of NCBI. So we have the EBI. They have comparable information. Actually, most of the information, there are some information in NCBI that are also in EBI, and EBI in NCBI. And then there are also other independent databases. Although most of these other databases are mostly specialized. So other portals such as the Exposé, uh, these other databases can also offer different analysis tools. Actually, NCBI, or rather, bioinformatics analysis tools, there are very many. There are so many bioinformatics tools, because a person can launch or can there are some researchers, or actually there are researchers that specialize on creating tools for analyzing bioinformatic or biological informations, and they launch their own websites for that. So how do we analyze the human genome? So another look at the workflow. So we can, how about if we want to know about the human genome? So first we need to choose, is it the individual? Of course, we want to know about their human genome, so get their informed consent, of course, and obtain raw data. You align. Now, basically, this obtaining part here is to sequence your DNA, and then we can compare. Based on the analysis, we can rather analyze it by aligning the reads to reference human genomes, and we can see the variants from here. And then afterwards, basically, this one is of course for the consent of the person you are using as your sample, obtaining the information by next generation sequencing. And then this one is the analysis part. So analyze the sequence of their genome by aligning it to the reference genome stored in the database. And then from based on the analysis, we can know if there are any variants and what are the functional significance of their variants. So these are the usual workflow for analyzing a certain human's genome. So bioinformatics represents a new field at the interface of the 20th century revolutions in molecular biology and computers. So in bioinformatics, we cannot remove the fact that we need the computers here. Computers as well as mathematical information, computer science and mathematics. So the use of computer databases and the computer algorithms to analyze proteins, genes, and the complete collections of the DNA is necessary. Because if you look at your sequences, these are 4 letters repeated in a certain sequence, in a certain unique sequence and we have long, very long repeats, so thousands to millions. Actually, the genome is in terms of number of sequence letters, we have billions. So we cannot analyze them manually. We need the computers. So a major challenge in biology is to make sense of the enormous quantities of the sequence data and structural data that are generated by genome sequencing project and proteomics. In fact, even for the human genome, which was fully sequenced way back 2003, there are still things there that we do not yet know. We can still find out more. There are many things that are yet to be explored. Even though it has been already here, we already know about the sequence 20 years, for 20 years, almost 20 years. Yeah, it's almost 20 years now. There are still many things that we have yet to know. So one of the most common things that or rather a field that is most commonly compared and or confused with bioinformatics is computational biology. So what is bioinformatics and what is computational biology? So let's go to bioinformatics first because this is what we are talking about before. So we have sequence structural analysis of the genes and genomes. So there is a limitation to bioinformatics. It's limited to sequence, structural, and functional analysis of genes and genomes and their corresponding products. They are often considered computational molecular biology. So it's a different side from the computational biology per se. So when we say computational molecular biology, we are just applying computational tools in molecular biology. Molecular biology is a bit different from biology as a whole. Molecular biology is a subset of biology. In molecular biology, we are looking at the different molecules that are involved. So the scope is a bit microscopic rather than biology, which is, well, basically holistic. Bioinformatics is the development and application of computational tools in managing all kinds of biological data, whereas computational biology is more confined to the theoretical development of algorithms used for bioinformatics. Whereas, let's look at computational biology. So, computational biology is a development and application of data analytical and theoretical methods, mathematical modeling, and computational simulation techniques to the study of biological behavior and social systems. So, in computational biology, there's more of mathematics here, mathematical models, as compared to bioinformatics. So, in bioinformatics, it's more of looking at the or rather analyzing. We can see it more of a tool. Bioinformatics, in my opinion, I see it more of a tool. It's a tool for analyzing molecular biology or information obtained from the molecular for the field of molecular biology. Whereas computational biology, it does not only concern itself with molecular biology, but the whole holistic view of biology as a whole. So in computational biology, there are models there that can monitor populations, predator-prey populations. There are also tools there that are used to explain certain metabolic pathways. Although bioinformatics can also be used in metabolic pathway analysis, but computational biology looks at a more macroscopic view of everything. So we have actually systems biology, which is another field which involves it's more of a meta view of the whole biological system. But anyway, so all in all, we can say as computational biology is a field, and it's a wider field than bioinformatics. So it's like IT and computer science. So they are very two sorry, they are two fields that are closely related, but they each has their own significant differences. So in informatics, basically information driven. So here it's bioinformatics. What drives bioinformatics is basically the information from biological information, analysis of this information, specifically DNA, RNA, and the proteins. In computational biology, what drives this one are the mathematical models. So mathematics is one of the main or important aspects of computational biology. Okay, so basically that is one of the some of the main differences of them. Although computational biology can also use information from bioinformatics. Okay, so we have three perspectives on bioinformatics. We have the cell, the organism, and the tree of life. Although I did say that bioinformatics deals more on molecular biology information, the molecular information of biology. The molecular information can give us an outlook into the cell, what is happening in the cell, the organism. Basically, for example, in this pandemic situation, we can use bioinformatic tools to know what drives the response of the of certain people, because in COVID, we are seeing a variety of responses, and we are seeing a variety of variants and the variety of responses to these variants. So we can look at bioinformatics tools to tell us about what's going on with our body, the organism first itself. And lastly, the tree of life. So this is mainly used in analyzing how one species related to another species in terms of their nucleic acid information. So the first perspective on the field of bioinformatics is the cell. So the emergence of molecular sequence of data drives this field of bioinformatics. So it's the main focus of the field of bioinformatics is to study the molecular sequence data to gain insight into a broad range of biological problems. So if you remember the central dogma, we have the DNA, which is the blueprint. It's the storage of the sequences, the genes. Basically, it has the information that the cell would need for it to live, to function. And this information is being encoded in the RNA. So not all genes are being encoded at the same time. So there are some genes that are silent, there are some genes that are active, and the active genes are being encoded by the RNA, and this RNA is then translated into proteins. So we have here levels of information in the cellular level. So we have what information is existing in the DNA, what information is being used, basically what are being expressed, the RNA data, what are the active, what are the inactive, because there are times that we have this information but we are not using that information. So the RNA is a better tool to know what is being used by the cell at this point in time versus what is not being used, or rather versus the DNA. And lastly, we have the proteins. So from the protein perspective, it's some people would say that proteins are much better in or rather proteins are a much better tool for the knowing what is actually being expressed, because the final product of the gene is the protein. So why do we have to go to the RNA? The main reason is because analyzing the RNA is far more easier than analyzing the translated proteins. But again, there are some proteins or structural information that we can have, although it's a bit tricky as of now. Until now, there are still some tricky questions or tricky things, or rather analyzing protein data or getting protein data are still a bit of a tricky business to have. There's still some difficulties on that. But in the protein data, we can know about actual structural information, how this certain gene, for example, going back to the previous example of hemoglobin, the hemoglobin, beta hemoglobin is existing, it's being expressed, but the structure of the hemoglobin, how it functions, we can only know about it if we know about the protein structure. Not the DNA, not the RNA, but the protein structure. How was it able to carry oxygen? So this is from the information on the protein. Now, we have dedicated databases for each level of the central dogma. So from the DNA, we have genomic, or basically nucleic acid, or DNA databases. And then for the RNA, we have the cDNA, the copy DNA. Basically, these are RNA that are reverse transcripted to DNA, express sequence tags, and the Unigene databases. And then for the proteins, we have protein sequence databases. Actually, aside from the protein sequence database, we also have protein structural databases like RCSB. And of course, this shows us, all in all, this information shows us the phenotype, or what is being, or what we see, what is being expressed, or what is being uh, what is happening, what phenomenon is occurring in that cell or in that organism. So there are three major public DNA databases. We have the EMBOL, which is housed at the EBI. EBI is the European Bioinformatics Institute. So each of them have their own links. They're actually integrated, or rather connected to one another. We can see some links referencing a certain data is in GenBank that is housed at the NCBI. This is the most well-known group of databases, actually. This is the National Center for Biotechnology Information. There are some data that are being housed, or references from GenBank to EMBOL, because perhaps it was originally submitted to EMBOL and then referenced by GenBank. Actually, the submission is basically based on the person who actually did generate that information. For example, your laboratory is capable of doing the sequence analysis or sequencing a certain gene. So it's up to the person who did that experiment, the group leader, to where they would submit it. But most of the people do it in NCBI, because most of the people do reference NCBI. So if you want, basically it's like you have a store, you want people to know about your store, so you go to a place where there are lots of people. So, and that so happens that it's NCBI. That is one of the most, actually these three. The other one is the DBJ, which is based in Japan. So this is the database of Japan. So again, some people do submit here. Now there's another databases that has been into a bit become more popular recently. That's the GSAID database. So GSAID databases. It's a nucleic acid, or rather, it's a DNA database. It's a genome. I think it also contains genomes, sequences. But this, I believe there has been some controversy, not exactly controversy, but apparently one of their vision is that they want to have a database that is not that can be used freely by people from countries that are, let's say, third world countries, or not so well known in terms of the biotechnology. So it's more like a rebellious, it's what I think, at least it's they want their own database. They don't want to rely on NCBI, EMBOL, or DDBJ. So they make their own databases, and they said that it's making the information available to the countries that are not as well known as, say, the US, the laboratories in Europe or in Japan. So I'm not really sure what they are trying to do with that one, but anyway. So that's where most of the COVID genome sequences of the Philippines, sequenced in the Philippines, instead of submitting them in GenBank, they submitted in GSA. And it's a more like you have to have an account manually approved by the people managing the databases in order to download or access the sequences there. So it's a bit of a hassle to actually do. But most of the sequences, say, in the Philippines was in there, at least for COVID sequences. I don't know why they had to do that. I believe that the information must be freely available to everyone, and why they have to diverge the sources. So that means if you can't find it in GenMac, you have to look at it for GSA, because the thing here is if GenBank, DDBJ, and MBOL can have connections, they can share some information, some data are shared between them. GSAID is a more, they don't want to share information with GenBank. So there are some sequences that are unique in the GSAID. The thing here is, GSAID is not as well known as NCBI. So there are less things there that you can find in NCBI, but less doesn't mean um, it's worthless. Sometimes there are some uh, critical data, critical sequences that might have been deposited in GSAID rather than in this one that you might need based on your test. And that is something that I'm not really agreeable with that, because why do they have to diverge the sources of this information? Anyway, so that's that. Things. Now, the second perspective, let's look at the second perspective. So the second perspective of bioinformatics is the organism. So bioinformatics tools can be applied to describe changes through developmental time, changes across the body regions, changes in a variety of physiological or pathological states. So for example, diseased versus normal. So we can use bioinformatics information, particularly the RNA expression information, in knowing if we have a disease or what could be a prognostic markers for diseases. And then for the third perspective, we have this the tree of life perspective. So we can know which organisms is much more closely related to one another. So one of the main basically practical applications for this one is in this pandemic, knowing the variants of the say the SARS-CoV-2 virus can help us pinpoint at least their genealogic information, where for some sleuths we can say that this variant originated in this certain area. So we can see here, we can map the progression of the variants, and sometimes you can also know where this particular variant came from. So that's one practical use of this tree of life. Okay, so we have some key bioinformatics websites. Again, these are the NCBI. GenBank is under NCBI. We have the EBI. It's a different source, but they have their own connection with NCBI also. And then we have the DBJ.